title of my talk is Life as a Sea Star Foster Parent. I'm going to be talking about the ecology, the life history, and the captive rearing of the sunflower star, Pycnopodia helianthoides. I graduated from the University of California, Davis with a BS in Marine and Coastal Sciences and had the awesome opportunity to do a postbac up at University of Washington's Friday Harbor Labs. And I worked with Jason Hoden on the first ever captive rearing program. And so I have a lot to share and so enjoy. Okay, so just a quick outline of how the presentation is gonna go. I'm gonna talk about what is a Pycnopodia. That's not a name that we hear very often unless you're in the science community. I'm gonna go through the events that led to the captive rearing, talk about spawning and larval culturing, meaning keeping them in a lab. We're gonna go through settlement and metamorphosis for the sunflower sea star, the juvenile experiments that we did, and the care for the individuals, and where we are now and what's next. So, Pycnopodia helianthoides, the sunflower sea star, is one of the largest sea stars, and they can have up to 24 arms as a fully grown adult. In this image, you can see the individuals that we had at Friday Harbor Lab, and these individuals were almost near a foot and a half from arm tip to arm tip, but they have been reportedly measured as large as 1 meter or 3.3 feet from arm tip to arm tip. So they're a pretty big sea star, and it can be hard to find a very large one, but they're super cool. And I have some videos, unfortunately with the Wi-Fi, they are a little laggy, but in this image, you can kind of make out they're over 15,000 tube feet on their aboral surface, which is kind of where the mouth is. And you can assume because of their, morphology that they can travel reportedly, it's been reported that they can travel up to 2 meters per minute when stressed. So pretty fast. They're like cheetahs of the tide pools, if you know. And here is that video. I don't know if it's looking very good, but I kind of zoom in onto the 2 feet, and there's thousands of them, and they use them for locomotion as well as capturing their prey.
It's working.
Say that again.
It's a little jerky, but it's working.
Awesome. And this is the aboral surface. So on this side of the sea star is what you would usually see when it's kind of crawling around. And it's covered in a lot of little protrusions. So they're called papillae and pedicellaria. They breathe through little kind of papillae, that's what they're called. That's kind of where they do passive breathing and oxygen diffusion so that they can, survive. And they have these little things called pedicellaria, which are kind of like little claws, and they look like they're the little spine-looking things on the surface, and they use that to keep themselves kind of clear of debris. And some sea stars, not this one, can even use them to hold on to prey, which is pretty cool. But you can see, because of all those protrusions, they kind of look a little fluffy. I think they're kind of the fluffiest of all the sea stars that I've encountered. If you're familiar with the tide pool critters, you'll notice that ochre stars or bat stars are a little rough or scaly, but these feel pretty fluffy, and it's really charismatic in my opinion. But yeah, so the sunflower sea star is a generalist predator here in the Northeastern Pacific. They live in subtidal areas, rocky intertidal and kelp forests, and they will consume a wide range of slow-moving and stationary invertebrates, although sea urchins, bivalves, and snails are notable prey for them. And studies have actually shown that they often elicit a dramatic escape response. So imagine the sea star is entering a tide pool. There's a lot of snails and urchins, and they sense that through chemical cues and immediately start running, or quote unquote running, and getting away from this predator as quick as they can, or as quick as tide pool critters can get away. But remember, these are pretty fast predators, so they're really efficient, and is one of the big factors that contribute to the sunflower sea star having an important function in kelp forest ecosystems. Now, I'm sure many of you have heard, but from 2013 to 2016, we observed a massive sea star wasting event that affected every known sea star in our region or asteroid species. And sea star populations kind of fell across the board. And you can see here are different kinds of sea stars exhibiting that sea star wasting disease. And it kind of, they start kind of looking a little deflated, a little white in color. They start amputating their arms. It's not good. And this is the sunflower sea star, unfortunately, wasting. It's not great, and ultimately they kind of away. And the sea stars that we had up at Friday Harbor were not free from that. There was wasting that got into our tanks. You can see they kind of look misshapen. They kind of start getting discolored. 11 of our sea stars, we lost 11 of our sea stars due to wasting in the last year. But luckily, if you want a silver lining, they took samples and they sent them to researchers to be able to better understand sea star wasting, because there's a lot we don't know. And you can imagine it's hard to study if these sea stars are pretty much, falling apart before your very eyes and eventually kind of disintegrate or melt, quote unquote. And so, yeah, so around the same time, we were experiencing a very intense marine heat wave, the most intense we recorded on our coast. Here in this image, you can see what we used to call the blob, quote unquote. And it kind of started as this, really intense sea surface temperature anomaly. So it got really hot around that area up there, around the Aleutian Islands, and that area north of our coast. And in July 2015, that kind of expanded. We got what I've heard called the son of the blob, kind of south along our coast through Baja California. And while those anomalies were occurring, we also had an El Nino. And so these marine heat waves took place from 2013 to 2015, and they kept the sea surface temperatures 2.5 degrees on average warmer than normal for 226 days. And so you can imagine that could be really, really stressful for all sorts of critters in our oceans, including potentially the sunflower sea star and other sea stars on our coast. And here you can see kind of the predictive occurrence of the Pycnopodia helianthoides, the sunflower sea star, from 2013 to 2020. And in the bottom, you'll see the red, the brown, the orange, and yellow curves. And those represent areas from Northern California down to Baja. And you can see by 2020, it's predicted that there was as much as it looks like 99% decrease in Pycnopodia populations. This study, Graham et al. 2021, took 48,810 surveys to quantify how the species was affected. And the study kind of showed that they're pretty much functionally extinct, meaning they're not doing their ecological role because they're not there. And there wasn't really a lot of evidence for recovery. And this is, cause for concern, right? Because they're pretty important for maintaining kelp forest systems along our coast. And just to kind of further explain what happened, these marine heat waves also affected the kelp forests. And you can see in this graph or this little diagram, in 2008, we had all this kelp coverage. You can see in the green, you have Van Dam, Point Arena Cove, Timber Cove, Fort Ross. And in 2014, 2015, and 2016, you can see that drastically fell. And that kind of coincided with the sea star wasting disease. And while kelp forests can usually be very resilient to short-term warming, multiple stressors, including climate stressors and ecological stressors, can tip kelp forests into urchin-dominated systems, which is kind of what happened. Without the sunflower sea star, in locations that had no other, important predator that can control these species, we had kelp forests kind of transition into urchin barrens. So urchins were released from predatory pressure. And So, these marine heat waves that happened affected the kelp forest, affected the sunflower sea stars, and it kind of just made this whole mess leading to these urchin barrens. And actually, something that wasn't appreciated with before this event, or from what I've come to understand, was the effect that sunflower sea stars had on urchin populations, specifically in locations that had a lack of sea otters or varied recovery of sea otters. And so, because they're one of the only predators that can efficiently consume them, we were still, able to see these kelp forests along our coasts. And now, without them, as many of you have seen yourselves, maybe there are urchin barrens all along our coast. And so that kind of led to the captive rearing project in partnership with the Nature Conservancy. So up at Friday Harbor Labs, Dr. Jason Hoden has been the lead scientist doing the captive rearing project. And the dual goals that we've had with this program is one, documenting important life history information, and raising them from egg to adult for potential release. And so there hasn't been a lot of research on their life history. And so that was the first goal. And to do that, we have to collect sea stars. So collections took place of adults through March through September of 2019 at the San Juan Islands in Washington State. And divers went down and collected individuals that were greater than 20 centimeters in diameter. It's not very clear how old these individuals were at this size. There isn't an easy way to measure that, since their anatomy doesn't really facilitate any way to kind of do that. And so we were kind of just guessing, or I'm sure there was educated guesses to figure out how big would be a good idea to collect. And so, we collected them. We had 28 individuals that were kept in 1 meter deep, partially covered tanks with flowing seawater. And to kind of help us better identify them, we gave them names. So we had Van Gogh, Charlotte, Crocus, Fanta, Deep Blue, and the divers had the opportunity to name them for whatever reason they chose. A lot of them because of their characteristics. Like Van Gogh kind of has these nice, beautiful patterns along its arms and whatnot. And so that was a lot of fun. We had 28 sea stars to begin with when I started working there about a year ago. And 2 were released due to potential wasting symptoms, one of which I would regularly see down at the dock. But 11 were lost due to sea star wasting in our tanks over time. And this is another video just kind of showing you the tank that we kept them in. And so, they were just in there. They're very friendly with each other, and they would sit around and kind of almost cuddle, if I say so myself. But that's the tank that we kept them in, and we would feed them mussels every 2 days, making sure they were happy and healthy. Yeah, so the next step is reproduction. So to the left, we have Clooney, which is a spawning male, and to the right, we have Charlotte, which is a spawning female. We can't tell if they're males or females until they've spawned, or unless you test for their reproductive gametes. And there's multiple ways to do that. But the way that we did that was injecting them with a 1 milliliter of a spawning and maturation hormone, which is called 1-methyladenine. And after 30 to 45 minutes, they'll spawn. And so that's kind of what we did. We would inject them, wait, have them spawn, and then unless we had already spawned them in the past, we'd wait to see if it was a male or female. And so we spawned a few of them early this January. We had about 3 males and 1 female that spawned. And so that was great. But other methods of spawning them include, or recovering their gametes, is to amputate them or create these incisions on their arms. That can be a little invasive. And so it was preferable to have them be injected and kind of just wait and allow them to spawn for us. And in case you're not familiar with broadcast spawning, it is a strategy that a lot of marine invertebrates use to reproduce. So they just spawn, meaning they kind of eject their gametes, whether it's sperm or eggs, into the water in hopes that they will interact, fertilize, and then that kind of creates a larvae. Depending on the invertebrate, it might be free floating, and that's kind of what we're going to get into. So we would collect their gametes, mix them together, know what crosses we had, so that if we in the future, if we needed to know, we could identify who were the parents. Here's another video real quick of the female spawning her eggs. You can kind of see, it might be a little blurry. Yeah, so next I'm going to talk about spawning, fertilization, and larval growth. And so here is a series of images. In A, you have the spawned eggs. B and C are a little bit closer, magnified version of those eggs. It took about an hour and a half for the eggs, once spawned, to be ready to be fertilized. And that's when we would mix them with the sperm from the males. And then image D shows a 2-cell stage, which is 5 hours post fertilization. And they grow until they become a mid-gastrula stage, which is image E. And you can kind of see that as a cell that's kind of forming. You can see the stomach is forming in there. And eventually, that gastrula will become a bipinera larvae, which is the first life stage in sea stars, or larval stage in sea stars. And you can kind of see it's kind of this little weird blobby looking thing, image F. And you can see the stomach, which is in red, which has some food in it. So it's been eating, and that is some algae that we feed the individual. And it's really cool because since they're so small, you can see through them and you can see the food that they're eating. And so image F, G, and H are all bipinera larvae, just different stages of them, and they kind of start getting a little bit more defined. You can see those kind of points around them. And image I is a late or a swimming bipinera larva, or Brachyloria larva, and this is 41 days post-fertilization. So it takes about 41 days to get them to a swimming Brachyloria larva, and that is kind of when they become competent. That's what we say when they're kind of fully grown larva. They're almost ready to metamorphose and become a juvenile sea star. But before we get into that, I will talk. I'll keep. There's some more images of the bipinera larva. We can look at them under a microscope pretty well, see if they're eating, see their growth, and measure. Part of what we were doing was kind of measuring them and doing a few experiments at different temperatures to kind of see growth rates at various temperatures, which maybe I can talk about later, but will not be the focus of this talk. But when we have the larva, we keep them in these kind of giant pickle jars, and we fill about 3 liters of water in them and keep them in there. And we have these larval stir rats, which kind of move slowly back and forth, kind of mixing the water, mixing the food that we throw in there, and keeping them kind of floating around without things kind of sinking to the bottom. And it's motor driven. So, we just kind of let it sit there, and it slowly stirs everything. We keep it in a big tank of water. They're usually kept at 10 to 11 degrees Celsius, and they were ready to feed 5 days after fertilization. So that's about the early bipinera larva. And they were initially cultured. When we keep them in those containers, we try to get 1 larva per milliliter of the container, and eventually kind of distribute them and spread them out into more containers as they're growing, so they have enough space to grow. And we feed them algae. So that's stuff that we cultured in the lab as well. So we, on top of raising these individuals, we have to grow and maintain all their food. For the larval stages, that is algae. We have on the left, the Noliella species, and on the right, a Rhodomonas species. And we would mix these up and feed them. And every 2 days, we would feed them and change their water in order to make sure that they had nice, clean containers. And that we were controlling when they would settle and metamorphose. So what is settling? Settlement is when a larva decides that it's ready to metamorphose, and they look and they assess their environment to decide whether or not they're ready to kind of metamorphose into the next life stage. And so in this case, a competent larva, meaning larva ready to settle, is on the top left. And that little guy kind of has you can kind of see it's a little blurry, but you can see the skeleton of the juvenile kind of hanging on its larval body. And now that larval body is kind of feeling around for space to settle. And so it's exploring. And once it's done exploring, it'll attach and decide if it's going to commit to settlement. And once it does commit to settlement, it'll start losing that larval body very slowly until all that's left is that little skeleton of the juvenile sea star. And you can see that on the right, it's on the top right, it's slowly, pulling in its body into itself. On the bottom left, you'll see that it's kind of completed that metamorphosis. And on the right, bottom right, you can see a bunch of little tiny newly metamorphosed sunflower sea stars. They kind of look like little flat pancakes in my opinion. And they're all sitting on that little bit of algae. It takes about 24 to 48 hours after a larva commits to the settler phase to kind of go through metamorphosis. Part of what Jason and the rest of our team were doing up there was figuring out the best kind of inducers of that metamorphosis. And one of the best ways that was discovered was putting them in containers that had kind of evidence or kind of a biofilm of adult sunflower sea stars, which is something we didn't know previously. Super cool. And so once they're metamorphosed, they're considered juveniles. And they start off as like less than 1 millimeter in size, which is kind of a speck. You kind of need a microscope for that. And so image A through N kind of shows different stages and different sizes of juvenile sea stars. The brown spots in the center, that's their gut. And you can kind of see that it kind of spreads out a bit into their arms. That is all kind of their gut for digestion. You can see that in images C and D, they kind of grow a few arms out of their kind of armpits, if you will, or out of their arms kind of laterally. Some individuals won't even have 5 arms. Some had 4 arms, which is kind of interesting. That's just kind of how they work. And in image E, you can see if you look a little closely, there's these little red dots on their arms. Those are little eye spots. And so it's really cool to see that the juvenile stage kind of has those eye spots, which kind of help them navigate. They mostly just sense kind of shadows and light. That's what we suspect, rather than the way that you would imagine an eye spot would work. But as you go through these images, you can see in G and H evidence of cannibalism, which we'll talk about. So at the bottom right in image N, we have individuals that are a little bit bigger. Don't mind that 5 millimeter in size marking at the bottom, that's incorrect. But they start acting like adults and they're kind of hanging out together, which is super cool. So let's keep going. So cannibalism.
Wait, Michael, can I stop for a couple of questions before you move on?
Yes, please.
Okay, so one question is, what is the larval skeleton made of?
That is a great question. It's not kind of like skeleton like bones per se, from what I understand. It's more kind of like that. It's like a early version of the body it's going to kind of move into when it kind of goes through metamorphosis. I'm not sure, like down to the exact, I'm not super sure what it is exactly made of, but it's just that, it's building that skeleton before it metamorphoses on its larval body.
Okay, and then another question asks, can you describe why the larvae choose the coralline algae to settle on?
Yeah, I'm not super sure as to the mechanism behind that, but coralline algae you see it in the tide pools all the time, and I think part of that is just because it's a nice little area that kind of keeps them safe. They has lots of stalks and they kind of cluster, and it kind of might be what's the word I'm looking for? Attractive for a little tiny organism that wants to make sure it can hide. But there's a lot of kind of reasons as to why it might choose to settle there, and it's not super clear.
I believe there's some new research about some cues being sent about settlement from the algae, but I don't know very much about that. Maybe Josie does. She asked the question. Josie, do you know?
No, I've just always heard that there was such a thing, but I've never really had it explained to me. It's always just been this thing that's out there that there is some chemical cue from the coralline algae, but it also makes sense that it's just a hard substrate for them.
Yeah,
Like,
yeah, and
like
it's
you
plenty,
mentioned.
and in normal times it's plentiful as well, so,
Right.
yeah. Okay, next question is, did you say dirt gut to describe what helps them digest?
I did not. Um, but it's that little brown area that you see on the juveniles. That is their gut, and that gut kind of spreads a bit into their arms as they grow.
Okay,
Yeah, that's
okay, and then how did you change out the water the larvae were in?
oh
How did
yeah.
you
So we forward filtered it or reverse filter. So we just filtered it through kind of a film, making sure that it was small enough that the larvae didn't pass through, and we would change that um, as they grew. But pretty much just kind of ran it through a filter and collected everything into a container. All the little juveniles into a small or all the little larvae into a container and cleaned them out, removing crud and because they were open to the air. And so after we were done with that, we'd put them back into their water and throw in some food.
Okay, and we have one question from your previous section, which was, why would you release diseased animals back into the wild?
So that is a good question. So I'm not super sure the reasoning behind why we did that, but ultimately we didn't know if it had wasting. So we didn't know if the individual was going to waste. It just wasn't looking super healthy. And because we don't know a lot about wasting, the idea was maybe they would do better if we put it back into its natural habitat right off the docks. Again, we don't know a lot about the disease. We don't know. I mean, yeah. So it might already be in the water, but that is a great question, a great thing to think about. That I am actually really curious and probably follow up with my previous mentor about that. So thank you for asking.
Okay, good. All right, that's it. We're caught up. Now you can go on to cannibalism.
Awesome. So cannibalism or sibling rivalry, whatever you want to call it. This is a really, really common thing to notice with these individuals. If we put all the juveniles together in one container, more often than not, we would find evidence of cannibalism. And so it became clear that we had to find ways to kind of separate them and keep them by size class to kind of prevent that from happening. But you can see in this image that the cannibal was sitting right under cannibal sitting right above a cannibalized sibling, unfortunately. But yeah, so we were observing that a lot and tried to avoid it as much as possible as they were growing. But there was a lot of evidence and a lot of observations we made of them eating urchins really early on, which is something that wasn't previously recorded. So individuals at this size, so sunflower sea stars at very early juvenile stages, were consuming juvenile sea urchins, which is really cool because that brings up a lot of questions about the role that that very specific stage of sunflower sea star might have in the environment, right? We know that adults eat sea urchins, but we didn't know previously that their juvenile counterparts did as well, which is super cool. And here is a graph that kind of shows just from our observations, every time we did a few checks, we would go through and we had all the juveniles in containers with juvenile sea urchins, and we would count how many we put in. We'd go through every few days and count how many individual juvenile urchins were still in the container. And we kind of had this very nice little graph that we made of how many juveniles were consumed per day, how many purple urchin juveniles were consumed per day by sunflower sea star individuals. And you can see that once they got past this 1 millimeter size threshold, they're eating very many. An individual sunflower sea star that is 5 millimeters in size on average was eating 3 to 4 urchins, which is really cool to hear, right? At that stage in their life, they're eating a lot of little urchins. But the issue we were having was below the 1 millimeter size range. So really early sunflower sea stars, we were having issues with what they were eating. we were having issues with mortality. It was a learning curve there. And so part of the research that I got to help with was figuring out what the best diet option was for an early stage Pycnopodia juvenile. And so this is right after they're done settling and after they've metamorphosed, trying to figure out how to efficiently and get as many individuals past that size range and figuring out what food they want to eat. And so we would put them in these little containers, these little jewelry boxes that we kind of made, putting one individual in each box. We did about 20 boxes. And we would give several individuals crushed purple urchins. So we would take very, very small juvenile urchins, crush them, and put them in a few containers. We took really early, recently settled sand dollar individuals, and then uncrushed, early settled purple urchins. And so that might be a mouthful, but here is a graph that kind of will help.
Start reading without any sign.
That will help kind of put that into, or visualize that. So we have a graph here, which is a standard box plot. You don't have to worry too much about what that means, but so we got on the far right, we got, or let me backtrack. So in the yellow, we have the initial diameter, the average range of initial diameter for all the individuals that we had in the experiment. And we measured after a few weeks their growth with different food options. And you can see that when given crushed purple urchin juveniles, it wasn't that big of a change between final diameter and initial diameter. And same was for the sand dollar juveniles. But when we gave them purple urchin juveniles, you could see that there was a drastic variation in growth between all the sunflower sea star juveniles. And so this kind of image on the right kind of gives you an idea of the kind of size growth that was happening. We got individuals that were 1 millimeter in size get up to 4-5 millimeters in size just from being fed purple urchins, tiny purple urchin juveniles. And so that is pretty cool news. We know they like purple urchins, even at that really early stage. And in this image, we have a bunch of Pycnopodia sunflower sea stars that are all live. And you can see those little rings. Those are the tests or the skeletons of purple urchins. And so this is kind of a little graveyard. They are quite the predator, even at really early life stages. And so this kind of a few images that kind of show our setup. We had them in, some seawater tables in plastic food grade containers. And we separated them by size class. We would go and measure them regularly, make sure that they were happy, count them, and just make general observations, because that was not something that was previously done. And when individuals needed special attention, we would put them in these beta tanks. And that is where we would kind of be able to find them fairly easily, because they are really small. And even a 6x6 container is pretty big. Sometimes we would lose track or struggle to find an individual and have to ask each other to get a second pair of eyes. And these containers kind of helped us keep track of individuals that didn't look very healthy. And so in our seawater tanks, we also had larger containers for the largest individuals. These right here are about 1 to 2 centimeters from armpit to armpit. And once they get to that size, we feed them pretty much exclusively little kind of tiny manila clams, because they're really good at opening them at that size. So that made things a little bit easier, because raising urchins as well as sunflower sea stars, takes up a lot of space. And so it's nice that they have different ways to eat, not just purple urchins once they get a little bit bigger. Here is an individual up close. You can see that, from my experience kind of looking at multiple sea stars, the sunflower sea star, even at really small size, looks, you can see the protrusions on its arms a little bit better. It looks a little fluffy, it looks a little softer than a lot of maybe the 6-rayed sea stars or small little ochre stars that you might see out on our coast. Here is a short video of one kind of crawling around. This one's about 1 to 2 centimeters in size. On the bottom right, you can see the little clams that we feed them, and it's kind of probably searching for food. They're really cute, very charismatic little critters. And here we got the 2-year-olds. But these images kind of show them at the stage where they were 1.5 years old. There's 12 of them. And in this image, they're about 8 to 10 centimeters in size. Here's a short little clip to you. And about 3 containers, separating them, 4 in one container, 3 in the other 2. And you can kind of see that they're all over each other. They're very cute. These individuals, we fed larger manila clams, as well as some mussels. And occasionally, if, in the mussels that we collected, if we had some snails or limpets, we would feed them to them, and they clean out every single shell. They definitely are the predators that we see when they are adults, even at these life stages. Here is a individual eating. You can kind of see that it's kind of has this little hunched shape. Yeah, and so here are a few more. And so as they were getting older and bigger, some of the challenges was finding out the best aquaria for them. And so this is kind of your standard like pet store tank. It was, relatively big. And we would throw in those plastic food grade containers in there and slowly allow them to kind of crawl out on their own. A few would come out immediately and kind of start exploring their new home. Others were very shy and afraid to even step out of what, you could say was their room for most of their life. But slowly but surely we got them into their new home. And we didn't initially allow all the individuals to interact freely. We put in this kind of almost like a film, like a plastic divider with a few holes in it to allow them to kind of interact without fully coming in contact, like full contact with each other, just to be sure and just to be safe. But here they are meeting and greeting these little cute individuals, and now they're besties. So this is kind of where I left the project, and we had them in all together in a container, and they were fairly happy. And now, as of maybe a month ago, the 2-year-olds have gotten a lot bigger. They're about 20 centimeters in size, as you can see here, and they kind of have gotten a lot more color to them. Prior to that, they were all kind of like a beige kind of color, which could have been because their parents were that color. But since we hadn't done a specific experiment to look at coloration, we aren't super sure if they are genetically the same color as their parents, or if they might express different colors at earlier life stages. So that's something that we still don't know, which is pretty cool to know. Anyway, so our first brood, we had 12 individuals. Those are our 2-year-olds. Our second brood, we have a total of 23 currently alive. And our third brood, which is the ones that we just spawned early this year, were 300 to 400 individuals, and about a third of them are over 1 millimeter in size. So that's good news, because that threshold, that 1 millimeter threshold, was at that stage there's really high mortality. And so knowing that about a third, about 100 of the new brood from this year are over that 1 millimeter threshold is really good. Anyway, when we did spawn them, we did have a lot of extra gametes that kind of got fertilized that we can't take care of. And so in this short video, you can kind of see, instead of just like letting them go down the seawater drain, we took them down to the dock. Since we had fertilized them and they were there, we decided to just toss them, pour them down the dock and allow them to kind of freely go into nature. But these individuals have a lot of things they have to deal with at the larval stage. They're plankton. There's the struggle of dealing with settlement and metamorphosis. So, we did that in hopes that maybe a few out of 1,000 will eventually, survive and become adults. anyway, just to kind of wrap up, the captive rearing program at Friday Harbor is really important because it's helping us better understand their basic life history. It's helping us better understand the different characteristics of their larval stage or juvenile stage, and figuring out the best way to raise them in a lab, or even if it's possible to raise them in a lab, which so far has been fairly positive. The next few goals are to potentially get to a point where we can do field-based recovery efforts in hopes of seeing if the individuals that we raise in a lab can actually, be viable out in the wild. And so that is the next step. So what is next? Like I said, can our lab raised sea stars survive in the wild? That is a really important question. And we're hoping to test that within the next year or so. Can we collectively raise enough for large scale wild reintroductions? It is very hard to get them past the 1 millimeter threshold. There's about 90 to 95% mortality when they're just recently settled. Very few survive. And so trying to further streamline that and efficiently raise them in captivity is important so that we can potentially do it on a larger scale. But continuing to identify the challenges for the early stage juvenile is critical. And potentially starting more captive rearing programs, if that is of interest, would really help, because there's only so much that one lab can do, and to better understand this species. So we're only at the start of what could happen for the sea star, and we're only at the start of what we know about the sea star. And yeah, so thank you so much for listening. And these are a bunch of names of a lot of people, individuals who helped with this project up at Friday Harbor Lab, and it wouldn't have been possible without all of them. So anyway, thank you so much for listening. I can answer some questions if there's any more.
Yeah, there's some more questions, Michael. My first question is, why do you think there aren't more people trying to do captive breeding? I mean, I know that there's a lot that the novel research you guys are doing, but I mean, with the need up and down the whole West Coast, why aren't more people trying it?
Yeah, I mean, well, one, it's really difficult to get a hold of those sea stars, right? They are critically endangered. You got to make sure that you have the right tools and the right individuals who can do the correct service for these sea stars. But also having a lab space, having the right equipment, that is a really important thing to do. And I think that through this first rearing program, we're able to kind of put together the steps for individuals or organizations that can have the space to have the captive rearing program. We're putting together those steps in order for it to be more streamlined.
So be more replicable.
Yeah, more replicable. And we're learning a lot from these individuals, and we're learning a lot from the sea stars that we have in the lab that could help streamline that in the future.
So in a presentation I saw from the Nature Conservancy recently, they talked about getting more adults from some of the aquaria in the Midwest because they came from the West Coast, just trying to increase that diversity issue. Do you know anything about that?
Yeah, so I know that genetically we might have a bottleneck if we're trying to reintroduce these individuals with like very few live adults. And so yeah, collecting individuals from aquariums or using individuals from aquariums that were collected off our coast years ago would be really helpful to kind of diversify in the the genetics of individuals, and not have a lot of siblings just reintroduced into the wild.
Okay, I'm going to just go down the list now with all the questions. So Christina asks how changes in salinity levels affect the species. Do you have any idea about that?
That is a great question. I'm not super sure about the specifics, and I can't say that we studied salinity changes with like any of the life stages yet. So that's something that is a really good thing to study in the future, and could be helpful in learning about. But I mean, Yeah, we don't really know much, or not. I don't know very much about that.
Did you guys have any problems with temperature? I've been hearing that some of the
Mhm.
the aquaculture efforts with the urchin are having problems with temperature. Did you guys have issues with variations in temperature?
Yeah, so actually, our project, we had, as many of you can remember, the heat wave that hit the North Pacific Northwest was really like, it was really damaging for a lot of reasons. But a lot, it affected us at Friday Harbor, and our water was a lot warmer. That was flowing through our tank systems and through the individuals, the juveniles, the very early, recently settled individuals. And we had a high mortality event that we think was due to that warming. So, we haven't done a lot of experiments to test for that, but just from that anecdotal evidence, it doesn't look great. But in terms of the ability for larvae to be able to adapt to that or survive different temperatures, we actually did do experiments testing the different rates of larval growth at different temperatures, including some really high temperatures, 17 to like around 18, 19 degrees Celsius for some of the larvae. And, I'm not saying that we have evidence for like hard evidence yet, but individuals weren't necessarily, falling like flies. So that's pretty good.
Good. All right. So another question, did you ever have problems with wasting disease in the juvenile stars you raised in the lab?
That's a really good question. It wasn't apparent because it wasn't, if there was, we didn't observe it, or we didn't know we were observing it. But there was high mortality in a lot of our juveniles, and it was at the same time that our adults were experiencing wasting. Although the water that was flowing through our tanks was, was filtered down to 1 micron, which is pretty nicely filtered water. That kind of kept our mortality low. But I don't know, that's something that wasn't very clear. And since they're so small, if they did get wasting and, went, just fell apart, disintegrated at a very, very tiny size, we wouldn't have known. Sometimes we were doing checks of the individuals and, find that 5 were missing, but there was no evidence that they were there at all. So that's a really good question.
Yeah. Okay, Bob points out that the skeleton is made of small plates of calcium carbonate. So there you go, from our community. And he says, great job, Michael, on your talk. Next question is, how hard was it to find the first adults in the sound, the ones you used initially for spawning?
That is a great question. So I actually didn't in that part of the experiment. So I actually can't speak to that. But I went out a few times on my own to kind of look in the tide pools, and I did find. So that's good news. So I can imagine they weren't as plentiful as they usually are, but they are still there, and they're still able to be found.
You froze when you said you did find, so we didn't get to hear what you did find.
Let me turn off my video.
You did.
I was able to find Pycnopodia out there on my own just by going out and exploring the tide pools. And so, they're still there and they're still easy to find. We found 28 individuals through the span of 3 months. So
it's pretty fair to say
yeah.
the population decreases the farther south you go down the coast.
Yeah,
yeah,
pretty much.
Yeah. Okay. Is there anything you can do to make them more resistant to wasting, I would assume, before releasing them? We don't understand wasting syndrome enough yet to answer that question.
That is a really great question. It'd be really great if we could do that, make them resistant to wasting. But there's a lot that we still don't know about wasting. And so the first step is to figure out what is happening when these sea stars are wasting.
Okay, Laura says, bravo, Michael, great job. Larry asks how citizen scientists can survey, which is excellent because we'll tell you exactly the answer to that at the end of this, because I think Joelle is part of our group, and she can make a pitch for our new program we have started. So Josie says there were plenty of sunflower sea stars in Alaska. I was just up in Kodiak and saw 4 juveniles in one little cove. When asked, folks said they see them regularly there. So that's excellent news. So they could be a brood stock potentially, right?
Yeah.
So Sarah says, thank you, really clear progression on their lives. How long do they live? How long does one live?
That is a great question. I actually don't know how long they'll live, but the ones we raised in lab, I can tell you, some idea, my hypothesis on that. But the individuals that we have in the lab, so the oldest reared individuals are about 20 centimeters in size right now, and they're 2 years old. And that is about the size that we were looking for, like the minimum size we were collecting at the very start of the project. So, the individuals that we collected were at the very least 2 to 3 years old in the best case scenario. So I don't really know how old they might live for, but yeah, I don't know.
Yeah,
that's a great question.
all right. That's what I remember when we were doing when we had the year of trying to figure out everything we knew about the problem, and the scientists at the time were telling us that if we had a bunch of juveniles out in the environment right now, it would take 7 or 8 years before we started seeing any impact from predation, just to size for them to grow to the size where they could do anything. But if you recall, I mean, sunflower sea stars were almost overpopulated before the wasting syndrome, and were like a concern for a lot of people because they were so voracious. So in other parts of the world, more of an issue for us, but certainly we're realizing now what an incredible role they played in balancing the ecosystem. All right, so Joelle asks, in the wild, is there a season that sunflowers spawn?
So we found that they were about ready to spawn between like around from November through early spring. Some papers said otherwise, so it's a little unclear from what I've been reading, because some papers suggest, previous papers before the project that we've been working on in Friday Harbor suggest that their spawning season can be from like April through May. So I guess it depends, and maybe that's changed because of wasting. It's hard to say.
Yeah. Okay. So Christina says, thanks for asking the question about salinity. It's certainly going to become more of an issue as D-cell plants are expected to release higher brine. Okay, so that's pretty much our questions. Does anyone else have any other questions to ask Michael? You can unmute yourself and ask him directly. If not, I'm going to ask Joelle to stay on for a second and tell everybody about the Sea Star project we have starting.
I think it might be critical, and maybe this is being done, to look at the causes of high mortality in the newly settled juveniles, because you could really up your production if you could solve some of those problems.
Yeah, you make an excellent point. A lot of the research that is now, like the focus now, is to figure out how to reduce that mortality rate. And so, that can mean having really nicely filtered water, making sure that their containers are very clean, making sure that they have, the right food. We know they like sea urchins. Maybe making sure that those sea urchins are very, very early settled sea urchins, because right after sea urchins metamorphose, they tend to lack the hard spines and hard external structure that they're just barely starting to grow. And so, I mean, personally, I've from my observations, I've noticed that when we have really plenty of recently settled urchins, the sunflower sea stars really ate them all. And so it might just be really hyper specific, or maybe there might just be some other selection pressure that we're not seeing.
But if that does get achieved, it sounds like there's a real interest in doing a lot with like what they do with abalone, where they spawn them at bodega and then they just give them to all the aquarium labs to grow. So if we get to that point, that would be a huge step. So if we can get it, I mean, it's nice to see your third cohort doing so much better than the others. So maybe we can get to the point where we start sending them out to just grow out in other facilities.
Yeah.
Okay, well, so then, thank you, Michael. That was super excellent, and you did a wonderful job. And it's really good to get a little bit more understanding about the process that's going on to try to solve this breeding issue, and get more of them in the environment. But before we say goodbye, I want to introduce Joelle if she's here. So Joelle is our lead for a new citizen science initiative Noyo Center has started, of which Michael is participating, called the Sea Star Project, which, because we saw the first sea star in Mendocino County. So the first sunflower sea star south of Fort Ord, I believe. We're going out and looking for more. So Joelle, you want to talk for a second? I have your graph of what we saw in the first round.
Okay, nice. Yeah, well, I see some of the STAR team members on this call, so that's good. And then, of course, anybody else who's interested in participating, I put the email address cstar@noyocenter.org in the chat, so you can reach out to us there. But yeah, so this is a new initiative with support from the Nature Conservancy, where we've reached out to our volunteers, staff, and interns to see who is interested in setting up kind of a community science project. And what we have is we have 7 teams up and down the Mendocino coast from Point Arena up to Westport. We go out at low tide events once a month, and we search for juvenile sea stars. And then while we're there, we do a count of other sea stars that we find and the types that we find. So that's why I was kind of wondering if there was a season for breeding, like, what size we're looking for. I know when they're small, they're hard to see. So it's wondering if as the summer progresses and we do more of these surveys, if we'll start seeing them. Even like 2 centimeters would be great. But yeah, so we just keep track of the locations, the species, and how many we've surveyed all together. And then we just do these monthly reports. And then after the last event, which is in October, we'll kind of combine all this information. And it'd be great to see some sunflower sea stars in our area. I know I've been keeping an eye on the iNaturalist app, and I'm seeing people are posting sightings of them up in like northern Washington and British Columbia. So it's a good sign. Hopefully they'll start coming down south again. But yeah, so cstar@noyocenter.org. If anyone's interested in joining our community science research initiative, we'd love to have you. We could have 8 or 10 teams up and down the coast. The more eyes out there, the better.
So UC Santa Cruz has a program called Marine, and we're uploading any information we find around sea stars to that. But we also are just tracking what other sea stars we're seeing. And so what's interesting is from this graph, the distribution. Now, we didn't give it, we're kind of dialing in our protocols. And so we didn't give everybody exactly the same, charge to go out and count all the sea stars, but people did do a lot of that. But you can see, like at Laguna Point, there was a cajillion bat stars, as there was on the headlands in Mendocino. So bat stars were really prolific, it seemed. But ochre sea stars at Westport and elk you know, I mean, it's interesting to see the distribution of stars in general. So because they are such a keystone species, it's interesting to get this information. So as Joelle said, if you're interested, email us and we will loop you in, but we basically are just going out and looking. So Larry, you're out there all the time. If you see anything that even resembles a sunflower sea star like Michael was showing in his presentation, take a photo of it, try to get an estimate of size and send it to us and we will send it up the chain, get it identified. We had a couple we thought might be, but they were not. What's really interesting is the way they grow the little arms and they, you know, kind of go out in a radial fashion, right, Michael? So that's something we're all learning about these guys.
Yeah, and something I'll point out is if we're finding them in the tide pools, they're going to be pretty far into the water, like really low tide area. And if we're seeing them at like 1 to 2 centimeters or larger, they're going to have more than 8 to 10 arms. And so you can rule out a lot of the 6th-rate sea stars that might, you know, be out there, because there are a lot of them out there and they're very cool, but they can be a little confusing.
Yeah, that's really great. From your presentation, you can really see how they get a lot of arms fast when they're little. So, well, thank you so much. I know we're at time now. Michael, that was awesome. Thank you for sharing all of your experience with the Friday Harbor Lab and all your work with sunflowers. We're super excited to have you on our team and look forward to doing more of this. I mean, now that we've got it in with those guys, we need to get some little babies down here and grow them out in a tank. All right. Okay. Well, I'm going to say goodbye then. Thank you all for joining us tonight. Again, if you are so inclined, noyocenter.org/donate, add $10 to the kitty, or better yet, become a member and support this kind of work. This is exactly the kind of reason why we need something like the Noyo Center on our coast. So thank you so much. Have a good evening. Michael, we'll see you out at the tide pools.